All right, and we are just about to get started here. Okay, so welcome to the second session of the Emory Theory and Modeling of Living Systems workshop on ecology and evolution. This one is on microbial diversity. Next month, September 24th, we're going to have the next workshop in our series. It'll be on taking biological theory seriously, plotting theory and data on the same axes. So I've asked in the chat for people to nominate their favorite recent examples of papers in ecology and evolution that actually do this, that have a quantitative theory that actually matches real data. So thanks to our administrator, Tiara Ward, and to the National Science Foundation's Physics of Living Systems Student Research Network for support. Please subscribe to our YouTube channel to get updates on future workshops. And please check out the Evolutionary and Ecological Systems Biology seminar series hosted by MIT that's usually at this time. I've put a link to that in the chat as well. A great series. Okay, so the format for our workshop today is going to be that we will have 20 to 25-minute talks with 5 to 10 minutes for questions afterwards. Please ask your questions in the chat window, and please go ahead and ask just as soon as the question comes to you during the talk. Don't wait till the end of the talk, because the YouTube stream is on like a 20-second delay from what we see. So if you wait till the end of the talk, I've already started asking the first question before you start typing yours in, and it may not get seen. So as soon as you think of it, please ask it in the chat. And if you start spamming or talking nonsense in the chat, we will go ahead and kick you out. So please be on your best behavior. After all the talks, we'll have another period for discussion at the end. So, feel free to just keep asking questions throughout, even about the previous talk, and we can collect them and get to them at the end. Okay, so this session is on microbial diversity. Why microbial diversity? Well, first of all, microbes are the vast majority of the tree of life. And they're the fastest evolving parts, and they have very rapid ecological turnover. So on the one hand, they are the most interesting intellectual problem in evolution and ecology. That is where evolution and ecological change are happening most rapidly. I'm going to go ahead and say that. If you disagree, go ahead and respond. They're also the most important practically, for related reasons. I mean, I think right now, I don't even really need to make the argument. Why are we all on Zoom? It's because of the emergence of a new virus. And, of course, we all know that most of the cells in our bodies are microbial, et cetera, et cetera. Hopefully this audience does not need to be convinced. But okay, so despite all this, we understand so little about microbial biology, and particularly microbial natural history. I mean, you take some of the best studied organisms. We're going to talk about E. coli. We're going to talk about Saccharomyces cerevisiae. We know so little about their natural history, about how they actually live. And the issue is really kind of a very simple one. They're small. They're hard to see. It sounds silly, but that's all there is to it. So the real challenge is, okay, how can we take the lenses that we have, in particular sequencing, but also other modern microbiological techniques, and use them to learn about how these organisms actually live in nature. I think we haven't really faced up to this challenge yet, but because we have these new lenses, I think we are poised for a real revolution in our understanding of the microbial world. And I think that the talks we have here today really point us in that direction. So with that said, let's get started with our first one. It'll be Erik van Nimwegen from the University of Basel talking about how bacterial phylogenies reflect complex patterns of recombination rates. So Erik, can you go ahead and share your screen, please?
Yes.
Okay, perfect. Take it away.
All right. Hi guys, hi everybody. First I'd like to thank Daniel and the other organizers for giving me the opportunity to talk about this work. So what I'll be talking about today is essentially contained in this preprint that I quoted here. And this project goes back a long time. I think it's, at least 8 years now. And the original idea of the project, we wanted to look at the evolution of E. coli in the wild. And when I say the wild, I mean this here. And so this here is actually at the shore of Lake Superior in Minnesota. So this is from Google Earth. If you zoom in, you see there's a parking lot, there's a landing where people can go with a boat. And the lab of Mike Sadowski went there about 15 years ago and took samples from the water, the watershed soil, and the inland soil, and then extracted clones of E. coli from these samples. And they send us about 500 strains. But for this analysis that I'll be talking about today, we focused on one plate of 96 strains. And actually what we did is we, apart from sequencing all these strains, we also, and that was sort of the main idea of the project, got extensive phenotype data. So we grew all these strains in many different conditions. And the idea was to relate their genome evolution to their phenotype evolution. All right, so one of the first things you do is you build a phylogenetic tree. So what I'm showing you here is a phylogenetic tree that we made from the whole genomes of a whole bunch of reference strains from the literature, from the database in black. All our strains are in red, and their names are just the well on the 96 well plate in which they were sitting. And you see a couple of green lab strains indicated as well. Now, what is already striking is that from this one spot, basically at one point in time, you find a very big diversity of strains covering most of the E. coli phylogeny and even covering an outgroup here that is sort of 8% diverged from all the other strains. All right, so we got this phylogeny, and because we wanted to model this evolution of the phenotypes we were measuring, okay, so the first question is sort of what does this phylogeny actually represent, right? How should we think about the relationships that this phylogeny shows? And of course, as all of you know, one of the sort of amazing things about biology is that every cell today on Earth is the result of a cell division, is the result of a cell division, is the result of a cell division. So these cells that we sequenced are somehow related with each other in some cell division tree. And I call this cell division tree the clonal phylogeny. And if it were the case that DNA sequences would only be transmitted vertically, that is to say, every time a cell copies its DNA and divides, then by looking at the DNA sequences today and reconstructing a phylogeny from those, we would essentially find the clonal phylogeny. And this would allow us, for example, to model the evolution of the phenotypes of these leaves along the branches of this tree. All right, but we also know for a very long time that not all DNA is vertically inherited. There are all kinds of processes that allow DNA to move horizontally. That is to say, DNA move from one bacterium into another bacterium and be homologously recombined into this bacterium. So the question is, what is the impact of this on the meaning of this phylogenetic tree that we built? And so as a first test, we decided to do something that has been done many times in the past, which is you cut your whole genome alignment into blocks. In this case, we cut it in blocks of 3 kb. We can use other sizes. And then we construct it using maximum likelihood a phylogenetic tree for each block. It turns out if you do that, every block has a different phylogeny. But in addition, if you do a sort of calculate the likelihood of every block's tree on the alignment chunk of every other block, they all basically confidently reject each other. So this is sort of the histogram of likelihood differences between, the likelihood of the preferred tree versus the tree from any of the other blocks for each of the blocks. All right, so it basically says every block thinks that it has a different phylogeny. Now, of course, it could be that these phylogenies are still very, very close to each other, okay? They're different, they're statistically significantly different, but they almost look the same. But this is also not really the case. So what I've shown you here is just the phylogeny of the 92 strain, the core tree phylogeny based from the core genome of these 92 strains. And what I've colored in each branch is for each branch, what fraction of the phylogenies from the 3 kb blocks contain that branch, okay? And so you see that actually 2/3 of the branches in this tree occur in less than half of the trees of the individual blocks, and half of the branches occur in less than 1/4 of the block, okay? So the trees that you get from the different blocks are not just sort of very subtle differences from each other, there are really substantial differences. So this all suggests that there are many different phylogenies occurring along the alignment instead of one dominant phylogeny. But if you do the following, if you take any set of a large number of blocks and you make a phylogenetic tree from this set, then those trees all look very, very similar. So what we did is we resampled many, many times half of the blocks in the alignment and rebuilt the tree. And now you see that actually 60% of the branches occur in all the subsets that we used, and 80% of the branches occur in more than half of the subsets. So this is not a new observation, all right? So in bacterial genomics, people have seen for a long time that there is, if you look at individual loci in the genome, there is a lot of evidence for horizontal transfer and recombination. But as soon as you build a tree from many, many blocks, these trees start converging to a common phylogeny. And also this phylogeny is consistent with phylogroups that people have, described in the past and so on. So I think for many people in the field, it seems rather obvious that this tree that you converge to when you use many, many blocks must be the clonal phylogeny, because what else could this be, right? The idea is there is some real clonal phylogeny, and this recombination is putting a lot of extra noise on top. So if you look at one block, this noise may dominate, but as soon as you take many, many blocks, then you sort of average out this noise coming from the recombination, and you recover the clonal phylogeny. All right. So I think our project sort of started from us asking ourselves, is that really true? And can we really convince ourselves that this is really what is happening in this data? Now, as it turns out, one nice way to actually sort of quantify recombination in pairs of strains, and especially close pairs of strains, is to look at the density of SNPs along the alignment of a pair of strains. So here I've taken two strains, F3 and E4, that are actually very close. And I basically compared them along the whole alignment. And in the blue line, I'm showing you the density of SNPs, okay, per KB or a couple KB. And what you see is that for most of the alignment, the SNP density is very, very low, except for a couple of places where all of a sudden the SNP density is much larger. And in fact, the SNP density in these regions is basically what is the typical divergence between pairs of E. coli strains. So for most of these genomes, these guys are very close compared to a typical difference of two E. coli's. But in these regions here, they are as distal as random E. coli's. And so basically, it's hard to imagine that this is anything else than what happened here is that these blocks correspond to segments that were homologously recombined from another strain of E. coli into one of these strains. Now, these blocks of high SNP density are reflected if you just take a histogram of, you go with 1 kb blocks over this pairwise alignment, and you just make a histogram, how many blocks have 0 SNPs, 1 SNP, 2 SNP, and so on. You see that this histogram is clearly made out of 2 different parts. So most 95% of the regions for this pair, their SNP distribution comes from a Poisson with on average 0.06 SNPs per kilobase, whereas the rest, 5% of the regions, roughly follow an exponential with mean 13.6 SNPs per kilobase. So what we did is we found that we can model all these histograms for pairs of species by a simple mixture model of a Poisson distribution for a fraction FC that was clonally inherited between the pair of strains, and a negative binomial distribution, which is actually really a mixture of Poissons with different rates for a fraction for the rest of the genome. All right, so, and what I'm going to show you now is if you take all pairs of our E. coli strains, I'm going to show you as a function of what was the overall divergence of each pair, so just what fraction of nucleotide differed for them, what was the estimated fraction that was clonally inherited for the pair. And so the first pair I showed you, as I said, it was very close. The overall SNP rate is 0.16%, and we estimate 97% of the genome was, of the core genome, was clonally inherited. But as soon as you start going to pairs of strains that are further apart, so this is a SNP rate of 0.6%, the clonal fraction has gone down to 50%, basically this part. And if you keep going, so this is 0.8% divergence, now you have 20% ancestral left. Here you have 10% ancestral left. And basically, as soon as the pairwise divergence is bigger than 1%, you lose all clonal inherited DNA in the genome. All right, so what we find from this pair analysis is that if you take pairs of strain, there are some pairs that are very close and where most of their DNA is clonally inherited. But in actually 80% of all pairs of strains are in a region where the entire genome has been overwritten by recombination and there is no clonally inherited DNA left. And so their pairwise divergence tells you nothing about their clonal distance because there's nothing left in their alignment that actually was clonally inherited. And moreover, even if you go to pairs of strains where there still is a substantial amount of clonally inherited DNA left, okay, so here I'm looking at pairs that have 40% up to sort of 80% clonally inherited DNA. It's still the case that of all the differences between them, 90% or more come from the recombined regions. So even for pairs where you still have a lot of clonal DNA left, their actual divergence is dominated by these clonal regions. And that's only not true for some guys that are extremely close together, okay? All right, so this is the analysis of pairs of strains. But we can also do some analysis where we look at the whole alignment at once. So, and there, basically, the main sort of method that we're going to use is we're going to look at the patterns that exist in biallelic SNPs. So to give you some relevant numbers, the core alignment that we have is about 2.5 megabase long. And of these positions, almost 90% are completely conserved. Every strain has the same letter. And there is 11% of positions that are SNPs, where there is polymorphism. However, almost all of the positions that are polymorphic, you find only 2 nucleotides. It's extremely rare to find 3 or 4 nucleotides. That is, of all SNP positions, 94% of them have only 2 alleles, okay? And you can do some back of the envelope calculations that show that this implies that the vast majority of these SNPs correspond to a single mutational event in the history of this position. all right? And so basically, this is a sort of a cartoon. So you see many positions are completely conserved. And then when you see a SNP, you only see two types, right? So a mutant type and a sort of a wild type. All right. So now these SNPs can tell you something about the phylogeny, right? Because if a SNP corresponds to a single mutational event, then if I see a SNP like this, right? So strains A, B, and C have one letter, D, E, and F have another letter, then whatever the phylogeny is at this position, it must contain a branch where strains D, E, F are on one side of the branch and strains A, B, and C are on the other side of the branch. And the mutation happened on this branch separating the two, right? Similarly, for this one, if you see E and F share this mutation G, then it means there must be a branch in the tree where E and F are on one side and A, B, C, D are on another side. And you can sort of combine information from such SNPs, right? So if you say, if I take these two together, now I can further refine the tree and say, look, I have to have both these branches. But a key observation is that you can also have pairs of such SNPs that are simply inconsistent with a common tree. So if you look at X and Z here, Z says that there must be a branch with C on D on one side and A, B, E, and F on the other side. And you cannot have that and have this branch in the tree at the same time, all right? So that shows that somewhere between X and Z, there must be a change in the phylogeny, okay? There must be at least one change in the phylogeny somewhere between X and Z. So you can use this to look at sort of how long are stretches along the genome that are compatible with a tree before you need to change the phylogeny. And you can look at, for example, also how many times minimally do I need to change the phylogeny as I go the entire alignment. And these kind of methods are actually very standard in the analysis of evolutionary, of sexually reproducing organisms. And so here we're simply applying them to our bacterial alignment. So if you do this for our E. coli data, you will find that actually the stretches of tree compatible SNPs are very, very short. All right. So typically it's, 3, 4, 5, 6 SNPs in a row, and then you get a phylogeny break. And it's extremely rare that you can get more than 20 in a row. All right. And if you look at what is the minimal number of times that the alignment has to change, as you, the phylogeny has to change as you go along the alignment, you find that it's at minimally 43,000 times, okay? So that says as you go along this 2.5 megabase alignment, you need to change the phylogeny at least 43,000 times. All right, so these sort of summary of some of the statistics that we looked at, and we looked at a whole bunch of other statistics, they suggest that this multiple alignment of the core genomes of our strains has been completely chewed up by recombination. For most pairs, no DNA was clonally inherited between them. There are more than 40,000 changes of phylogeny along the alignment, and simple back of the envelopes calculations also show that given the size of these recombined segments that we can estimate from the closed pairs, as I showed you a little while before, each position in the alignment has been overwritten at least 100 times, if not 1,000 times. Okay. So given that, the question is, why do you get a consistent phylogeny when you use many blocks, right? Because we saw in the beginning that if you build a phylogeny from any large subset of the blocks in the alignment, you always get the same. You always get the same phylogeny, very close phylogeny. So if recombination is so dominant, how can you get such a clear phylogenetic signal? Right. So to explain that, what I did here is I went to the database of the 1,000 Genomes Project from human, and we sampled a random set of human genomes, took autosomal chromosomes, and basically did the same thing. We built a core tree, and then we resampled large numbers of loci and rebuilt the tree. And you will find that you will also get a robust phylogeny. All right, so you get very robust phylogeny where most of these branches occur in most of your resamplings. And they also have patterns that make sense, right? So individuals that have the same geographic ancestry, they cluster together in this tree, all right? So why is this happening? Well, we know that there is no clonal phylogeny for human sequences. There's no such thing, right? I mean, there's no, every organism is a mixture of two parents and not just one cell division. And so, so we know that there are many different phylogenies occurring along the alignment of the human genomes. But we also know that there is population structure, all right? So not any two humans are equally likely to recombine. Some lineages are more likely to recombine than others. And that means that these phylogenies that occur along the alignment are not uniform random. Okay, so recombination is not just noise. Recombination has its own structure. And so what the core genome phylogeny represent is essentially an average of this distribution of phylogenies along the genome. And the fact that it becomes highly robust is just because if you average over enough loci, your average becomes very robust. All right, now, so in the final minutes, I just want to mention how you can use these biallelic SNPs to learn about the distribution of phylogenies along the genome and to learn something about their structure. So for example, you can take SNPs of the following kind. You can say, let's look at all SNPs where only two strains share the SNP. And one is strain A1 and the other is another strain. So when I see a SNP of this type, it means that at this position in the alignment, the strains X and A1 must have been nearest neighbor in the phylogeny, and the mutation occurred on the branch going to the ancestor of X and A1. Now notice if there was a single phylogeny, then A1 could only occur in such two SNPs with one other strain X. But in reality, what we see is that A1 occurs with many other strains in two SNPs. So we find two SNPs with more than a dozen different strains for A1. If the population were fully mixed and everybody was recombining equally often with everybody else, then you would expect this distribution of pair SNPs for A1 to be roughly uniform and have equal numbers for every other strain. But that's also not what we see. A1 occurs in a pair much more often with some strains than with other strains. Okay, so this reflects the population structure that's there. And so you can, for example, look at the distribution of all pair SNPs. And you see that every strain tends to occur in pair SNPs with lots of other strains, but with a very non-uniform distribution. Some pairs are much more common than others. If you make a histogram of the distribution of pair SNPs, you find that these distributions have actually long power law-like tails. So that says there's no typical scale. There's neither a single fully mixed recombining population. Some recombination events are much more likely than others, but there is also not a clear scale at which you can divide the population into subpopulations. This is not just for pairs. You can play this game for triplets, for quartets, for dozens, for any number. And we see that all these distributions have long tails and that there is some information in how the exponents of these distributions changes as you go to larger numbers. That is to say, a 12 SNP tends to have occurred further in the past than a 2 SNP because it's shared by more strains. And the distribution of these pairs of 12 SNPs clearly has a higher exponent than the 2 SNPs. All right, so maybe I'll skip this. Another way that you can look at this is you can look for each strain and you can ask how many different pairs does it occur with, or how many different triplets or quartets and so on. So you can sort of make an entropy profile that says how diverse is each strain, the phylogenies of each strain as you go to groups, larger and larger groups from 2 to 3 to 4 to 5. And what I think is interesting that if you make these entropy profiles, they basically look different for every strain. It sort of suggests that the recombination rates of every lineage with the other lineages are different for every lineage. All right, so with that, I've come to the end. So the key things that I want you to take away of this is that for most pairs of strains, none of the DNA in their alignment stems from the clonal ancestor. All has been recombined at some point in history. Even for closed pairs, more than 90% of the divergence is induced by recombination. The clonal phylogeny of the species, we don't believe you can reconstruct anymore. It has been overwritten by recombination too much. But what the phylogenetic structure that you see represents is actually the population structure. And this population structure looks complex. There is not a single recombining population or clearly separate subpopulations, but there is a long continuum of relative rates. All right, so that's it. And thanks for your attention.
Thank you very much. Let's jump right into the questions. I'm going to go a little bit out of order here. So Purushotam Dixit asks, could that structure be spontaneously generated by divergence-based suppression of recombination?
Yeah, so that's a very interesting idea, and I know he and some of his collaborators have worked on this. So this is, I mean, what is so attractive about this idea is it's very beautiful, right? So the idea is that homologous recombination needs primers to work. And so as two strains get more and more diverged, the efficiency of this homologous recombination process goes down, and it maybe goes down exponentially. And so maybe you can get sort of this kind of different rates appear automatically because guys that are close, it's easier for them to homologously recombine than guys that have diverged. Now, my impression is that most of the strains in our collection, they're all within 3% divergence, with the exception of this outgroup. And we see very, 3 or more orders of magnitude differences in these relative rates with which we see these different SNP patterns occur. And it seems implausible to me that just 1, 2, 3% divergence is enough to make such large range of different rates. So, I mean, yeah, we cannot exclude that it's that simple, but I would personally sort of think that it's maybe not that simple and maybe other things going on.
Good. All right. Jane Charlesworth asks, I'll paraphrase here, that, could you say that basically the problem is that even rare recombination is enough to erase any traces of the true underlying clonal history?
I guess there's a question of the definition of rare. I mean, it's as rare as you see it is, right? I mean, so the problem is that I cannot really summarize the recombination rate with one number because, as the data suggests, it varies over orders of magnitude between different lineages, and it's probably even the case that if very close strains recombine a lot, we can't detect those, right? So we're probably underestimating the rate of recombination between very close pairs. So, I mean, okay, I don't know whether you want to call it rare or not rare, but it's enough to have overwritten everything many times over.
Good. Mahan Ghaffari asks, how sensitive are the reconstructed phylogenies to the choice of substitution model? Is there a lot of heterogeneity among sites and substitution rates?
Yeah, no, I don't think that matters at all. I mean, you can play around with all these things and you may get subtle changes, but I think the global phylogeny that you get is fairly robust with all these things.
Good. All right. So I have a question that I want to ask. Like, you can do hierarchical clustering on any data. When we do it on population data, usually it's because we believe that these hierarchies reflect a true historical tree. Should we try something non-hierarchical here? Because if we don't think that necessarily a tree is producing this diversity?
Yeah, so this is all right. So your question is, maybe there is not one tree, but maybe summarizing the structure by a tree is still the most relevant way of summarizing what the structure is. I don't know. I don't think so, but I don't have anything better at the moment. Okay, so so I like these entropy profiles. I like looking at these SNP distributions, but I think we really need something new where we really find a way to represent the entire distribution of SNPs by a lower dimensional sort of representation, and find the low dimensional representation of the entire statistics that you see. But I've not been able to come up with such a model yet. But what I do think is very important is that taking this tree and then modeling evolution as if it occurs along the branches of the tree, that's really not correct, okay? Because that's really not what's happening.
Well, that's a perfect place to end it. So Erik, can you stop sharing your screen? Yeah. And check out there's some more comments in the chat that you may want to check out. All right, so next up we have Jacopo Grilli from ICTP talking about laws of diversity and variation in microbial communities. So Jacopo, can you go ahead and share your screen?
Yes. Can you see?
Good. Yep, and you want to just go to presentation mode.
Okay, great. So thank you very much for the invitation, and it's a pleasure to be virtually there. So before I start, I want to spend a word on where I'm physically at. And I work at ICTP, which is a UNESCO research center in Trieste that has a double mission to do good science, but also to foster science and excellence in science in developing countries. So we organize many activities. We have many visitors from everywhere in the world. If you would like to get involved when this pandemic will be over, if it will be over, please come here. But in the meanwhile, we have this school coming up in a few months, organized by Samuel Levin, myself, Matteo Marsili, and Antonio Celani on quantitative approach in ecosystem ecology. So this is the end of the advertisement. Sorry to waste 1 minute of my time for that. So the title of this workshop is very much aligned with the question that keeps me up at night when my son doesn't want to sleep, and is what determines microbial diversity. So this is a very foundational question in ecology, because it's basically equivalent to ask why does ecology exist in the first place, and I will not even try to answer to this question during my talk. I just want to mention that this has sort of been the most controversial issue in the history of ecology, and depending on the decade you were working on, you will have thought of one of the two sides of this battleground. And basically this fight is between two different interpretations of what are the leading forces determining diversity. One is that diversity is determined mostly by random processes. So basically species or types are there because they didn't have time yet to get extinct. And versus necessity, they are there because they occupy a very precise niche, they have a precise role, and they are there because they could not be there. So as I said, I will not touch this question directly. The question I will try to attack is mostly about variation of species. So, and is related to what are the mechanisms that determine the variation of species presence and abundance. And in the microbial, but also in the non-microbial world, you can name many, many different ecological forces that for sure have an effect on species presence and variation in abundance. So the question I want to try to understand here is, can we sort of have a quantitative understanding of how much they affect the variation we observe, and what are the relevant observables to understand this effect? Now, one important caveat to this is that there is no universal answer to this question. The answer to this question depends on what spatial, temporal, taxonomic, phylogenetic scale we are looking our community at. So at the different scale, mechanism will differ. And so this is a sort of disclaimer that whatever I'm going to say, it's something that holds for the spatial, temporal, and taxonomic scale I'm looking at. Great. So in general, I think the main difficulty in microbial ecology is that we are dealing with very high dimensional ecosystems where we have many, many different species, many types, many conditions. And when we deal with high dimensional data, we need somehow to do some sort of dimensionality reduction. So I think that there are sort of three ways to approach this. This is not exhaustive. There might be many more. But one way, which is sort of the most standard way, is to somehow try to aggregate species or community and do a dimensionality reduction, looking at what aggregation of communities explain or species explain most of the variation. Another approach is to simplify the setting. So try to understand which mechanisms determine variation by looking at well-defined experimental setting with few control conditions. There is a third approach, which is which is like focus on the typical properties of species variation. So to embrace the variation, don't try to reduce it to a smaller dimensional manifold, but to look at what are the typical properties of species variation. And this is the approach I'll take, which has a long history in ecology. It was pioneered by James Brown in the early 90s, I believe, and it's called macroecology, and is the idea of looking at communities as some sort of statistical ensembles. There are many, an increasing body of work using macroecological approach in microbial communities. And so the work I present here is sort of inserting this in this direction of work. Great. So the two questions that I more explicitly try to answer today is what are the invariant properties of the variation of species presence and abundance, and what are the mechanisms determining this variation? So I'll first discuss a few statistical properties that are regular across biomes and use them to define a null model to generate microbial community in silico, showing that this null model reproduces other statistical commonly studied properties. I'll compare variation across communities and across time in the light of these statistical properties. And then I'll use these to try to understand something about mechanisms. But first, I want to spend a few seconds on the data. You are probably much more familiar than me with the data, but I just want to say that there is a huge experimental and bioinformatic pipeline to go from samples to the data we use. And there are many sources of noise, of technical noise in this process, and I'll make a simplification which might be very strong. So the simplification I'll make is that all this noise, this technical noise can be modeled as sampling noise. So for sure, this is a huge source of technical noise. For sure, we are sampling a small fraction of the individuals present in the communities. The thing you might disagree with is that this is not the main source of technical noise. I don't have time to go through the details, but we can discuss online or offline of what one can do to show that this is actually the case, that most of the variation can be modeled as, technical variation can be modeled as sampling noise. So that means that basically we want to go from a set of frequencies, the fraction of species, of individuals of species I in a given sample, to discrete reads in one sample. And this is just multinomial sampling. And if you are interested in a statistical ensemble, we have a distribution over these frequencies, and we go through multinomial sampling to a distribution over the data that we can observe, which are given the sampling depth, the sampling effort that we use in our data. So our goal is to infer properties of this distribution of the true abundances, and we have access only to the data we have, so to this distribution piece. So this is something we should take into consideration while we look at the variation in the data. Great. So what patterns can we look at? Well, the most studied pattern in ecology is the species abundance distribution, which is just a distribution of abundances of species in a given community. So you can look at what is the fraction of species that is present in more than a given number of reads in a given community, and you find a distribution. And there is a large body of work trying to understand what is the right statistical model for this distribution and what are the mechanisms that produce these statistical model as a result. Now, if you repeat this for another community, you find some sort of common curve with some dispersion. And the important point I want to make, so this is like very much studied. This is studied from microbes to tropical forests. Now, there is one point that I think it's not well discussed, is that there are three sources of variation determining this pattern. Where one is actually very well studied, which is the effect of sampling. We are still sampling a finite number of individuals, but there are two different sources of variation that both contribute to the variation in species abundance distribution. One is the variation of abundance of a species across communities, and the other one is the variation of abundance across species. So to make a cartoon example, we could have these two different patterns, where two extreme patterns. On the left, one case where species have always the same abundance across communities, but they just differ between species, versus a case where species abundance is identically distributed across species, but they vary across communities. They will produce exactly the same species abundance distribution. So can we disentangle these two sources of variation? Well, the simple things you can think of doing is to look at the variation of abundance of a given species across communities. Now, there is a technical issue here, which is that different communities are sampled with different sampling depth. So you have to take that into account. If the species is abundant enough, you can forget about that, and you can basically look at the frequency, and you can look at what is the distribution of the frequency across communities. And what you find is that across very different biomes, this distribution is well described by a gamma distribution. Now, this is true for very abundant species. You can ask, is this true also for rarer species? Now, to do that, you have to remove the effect of sampling errors. And a simple way to visualize it is to compute from the data the moment, something related to the moment generating function, do a proper rescaling, and basically what you get is a prediction assuming that the data are gamma distributed, and this is what you find. So the black line is the prediction if this distribution of abundance across communities gamma distributed. The colored points are the bin data, and the gray lines are individual species. So you find that the gamma distribution describes well also the variation of rarer species. Now, there is one unintended consequence of having a gamma distribution describing variation. And the consequence is that the probability of having a zero, so the mass in zero is zero for a gamma distribution. So it's like if all the species were always present. Now, of course, if you look at the data, you find that most of this, the vast majority of the species are not always present. So how that is possible? Well, for sure, one source of absence is also sampling, right? So if you are sampling a finite number of individuals and a species is rare enough, there is a probability that that species is absent. Right? So what you can do is that if you know the parameters of the gamma distribution, you can estimate from the abundance of the species, you can estimate how many times do you expect a given species to be absent, or actually how many times you can expect a given species to be present, right? And so you can test with this the data, and it works well. So you can actually predict the fraction of samples where a species is present. If you want to do something more rigorous, you can do model selection with a model with true zeros and not true zeros, but zeros only due by sampling, and the model without zeros is typically favored for the vast majority of the species. Good. So what I've been saying so far is that if you look at a given species, a given OTU, and you look at the variation across communities, this variation is well described by a gamma distribution. Now that means that for each species, I have two parameters, which are related to the mean and the variance of this distribution, and they characterize the variation of that species across communities. A natural question to ask is, are these two parameters independent across species? And as you might expect, the answer is no, they are actually related. And in particular, the variance scales linearly with the mean, which means that the coefficient of variation doesn't scale with the mean. And this relation is known as Taylor's law, and this Taylor's law holds with exponent 2. So effectively, there were two parameters per species, but they are related. So there is one parameter per species, which is the mean abundance. What is the average abundance? So now the natural question to ask is there any regularity in the distribution of abundance across species? So what you find is that the distribution of average abundance is log-normally distributed across species. Again, for all the volumes I looked at. Now there is one caveat here. So again, we have sampling. So if a species is rare enough, you will never observe it. So there is a lower cutoff of the log normal, which depends on the sampling effort, on how many samples you have collected and how much you have sequenced them. And that determines what is the minimal abundance you can detect. Okay. So there are these three statistical patterns, laws in quotes, and one can use them to generate a null model, to generate microbial communities in silico and test whether they have the same statistical properties of empirical communities. So basically, putting together these laws, you can write what is the probability that in a community with a given total number of reads, you're going to observe a given number of reads belonging to a species with a given average abundance, or if you take a species at random, what is the probability that that species is present with a given number of reads? In this sense, it's a null model. And with this null model, you can compute quantities. So for instance, you can compute what is the expected number of different species that I observe in a sample with a given total number of reads, capital N. Now, this prediction is a prediction in the sense that all the parameters that enter in this calculation here are set for each biome once you have observed the three macroecological laws, right? So looking at the coefficient of variation of the log normal and the parameters of, the parameters of the log normal and the coefficient of variation of the gamma distribution, you have set all the parameters. So you can look at this prediction and test with the data. So for instance, this is the number of observed species versus the total number of reads. The black line is the prediction. The colored points are the bin data, and the gray points are individual data. And again, this is not a fit. I'm not fitting any parameters specifically to this curve. And it works very well, both in predicting the trend and in predicting the value. You can look at the species abundance distribution, and the shape is well predicted. It's also predicted well the effect of increasing sampling. You can look at the distribution of occupancy, so the probability that a given species is present in a given fraction of the samples. Again, it works fairly well. You can look at the relation between occupancy and the average relative abundance. So what is the fraction of samples that a species with a given average relative abundance is present in? The prediction works like that. So this is a reasonable good model which agrees with many statistical properties of these communities. Good. So next step is to compare variation cross-sectional variation across communities, cross-sectional data versus longitudinal variation, so variation across time. Now, of course, we know that it's not the same, but we can ask whether the same patterns hold, right? And the answer is yes. So we still see the same patterns, and we can also check whether the communities generated in silico match the other statistical patterns, and it works very well. So again, the statistical properties match the data. Great. So at this point, one can stop and ask, well, is this due to some technical properties? So am I finding the variation, these regularities, because basically I'm always sequencing in the same way, I'm always collecting data in the same way? Well, one can check that with technical replicates, and the answer is that the variation with technical replicates, you can explicitly check that, and you see that most of the variation is not technical, it's biological. So these properties are somehow related to some biological factor determining them. So if you believe then the question is, okay, what is the biological factors that determines this pattern? So here I want to present one model that is in agreement with the data. It takes the form of a stochastic differential equation where you model the variation of abundance over time of a given species. And there is a deterministic term which corresponds to logistic growth and a term corresponding to fluctuating environment. So this simple equation reproduces the gamma distribution as stationary distribution. And if the parameters are chosen opportunely, also reproduce the other patterns. In particular, if the parameter sigma is constant, Taylor's law is recovered, and if the parameters, the carrying capacity is log normally distributed, then you get a log normal distribution of average abundance. You can also check the models on dynamics to check whether they reproduce dynamic patterns. And in principle, all the non-stationary properties are determined by the transition probability. So the probability that if a species is present with a given abundance today, it's going to have a given abundance tomorrow, right? So you can look at the moments of this distribution, aggregating in a proper way over species, and you find these patterns. So the colored points are the data, and the gray line is what you would predict if you scramble the data over time. So you see a discrepancy, which means that the data carry out information about dynamics, and you can check whether the model predicts those, and you find that yes, the model predicts those. Good. Now, a few months ago, I was checking bioRxiv one morning, and I found a preprint whose title was explicitly stating the same results that I have. And typically, you are upset because you say, "Well, I'm going to be scooped. I'm going to have to fight about this." In my case, I was relieved because it was reaching exactly the same conclusion using different data and a different approach. And so this paper has been recently published and basically has a very similar claim to the claim I just conveyed to you. Good. Okay, so something that I always ask about, and what about neutral theory? Can it reproduce the data? So it depends on what you mean with neutral theory. So if you mean plain vanilla, extreme neutral theory, well, all the per capita rates are equal for all species, no. It cannot reproduce the data because it doesn't reproduce the variation in average abundance. You can have a meta-community model where you have immigration rates varying across species and equal birth and death for all species. And that model also can be falsified because it doesn't reproduce the correct exponent for Taylor's law. You can actually reproduce the stationary properties, fine-tuning the birth and the death rate, but it doesn't work for dynamics. In the paper I just cited and other papers, they do that analysis and show that demographic stochasticity is not consistent with the data. Great. Okay, so to be somewhat provocative, this is a natural question that I totally agree with, and you might have at this point. So is this some technical artifact? When you see that in genomic, usually you think that is the case. So I want to open a box just to mention an old paper by Erik, where you actually find remarkable regularities in genomics, in the functional content of genomics, just as an example of something completely different at a completely different scales, where you have some robust pattern that is not a consequence of sequencing artifacts. So this, it's easier to answer. Now, the hard question here is this the real deal, right? And my answer is no, I don't think that this is the real deal. I think this is more the ideal deal, right? In the sense that, so first of all, this is something that is valid at and this is, I think, very important, is valid at a given taxonomic resolution. So if you change the taxonomic resolution, both fine graining or coarse graining, you are going to find something different. And also, this fails in reproducing other statistical properties. For instance, and I'll go fast on this, it fails in reproduces the correlations, the typical correlation of species. So this is not the real deal. And on the mechanistic side, well, okay, let me jump to the conclusion. So what is the take-home message? So since you are already home, what should you tell your kids? So one is that there are quantitative regularities in species presence and abundance. And so there is null models that can allow you to generate microbial communities in silico, and you can use that to test whatever hypothesis you have on the data. The other one is that the regularities of this data reflect regularities in ecological process. So there is an effective or mechanistic, you can choose, model that reproduce this data. And somehow the forces that are completely different across biomes, the high dimensional space of condition is somehow recapitulated by environmental force, so environmental noise. So the real deal is sort of opening the box of this environmental noise and look what is inside the box, in my opinion. And with that, I conclude. Thank you very much.
Thank you. Yeah, great. I still can't believe those patterns. All right, so there's a lot of questions along similar lines about kind of your point 2 here about the kind of underlying mechanistic model. So Ilya Neyman asks, can you say again, where do these laws come from? Why are, say, the parameters distributed, log normal for carrying capacity? What's the underlying model?
Okay, so for the log normal of carrying capacity, I have no idea. So I don't know where it comes from. So I don't have any explanation of understanding how that emerged, except waving my hand and saying multiplicative something.
Yes, and that, yeah, some questions
but
along that.
I don't have any model for that. So what I can say is just you have to parameterize the model in that way to reproduce that patterns, but I don't have an explanation for that.
Yeah, so it's specifically about the kind of, multiplicative noise thing. Yep, Eric asks, is that the key ingredient? You say yes. Sri Biswas asks, can that multiplicative noise be pretty generic, or does it need to satisfy any special properties?
Okay, so in general, if you want to reproduce any stationary distribution, so if you are just interested in this pattern, you can write infinite. Actually, you can write all the models that reproduce a given stationary distribution, and what you have to tune is the deterministic term with the multiplicative noise. So if you give me a deterministic term, I can give you what is the multiplicative noise that corresponds to this. So in general, there are many other models there are infinitely many models that reproduce this pattern here. That's not true for the non-stationary data. If you fix the deterministic term, then this is the only choice of the multiplicative noise that reproduce the gamma distribution. So if you change that, you get a different prediction.
Good. All right. So Silvia De Monte asked a question that I'll paraphrase as basically wanted to probe further the thing you said about how everything could be captured just as sampling noise. Can you briefly explain why that works?
Well, you can, okay, so what you can do is that you can look at the variation that you observe in technical replicates, and you can check how much, you can check how much of the noise is captured by sampling noise, or actually whether the patterns that you see in the variation are similar to the ones you would expect from sampling noise, and you observe that. So in particular, the point is that sampling noise has a particular effect, that it's the magnitude of the noise becomes more and more important as you decrease abundance, as something becomes rarer, and this is what you see in the data. But I mean, the important point to state it clearly is technical variations, once you take into account for sampling, is small compared to biological variation.
But I mean, I think the question is, the technical variation all follows all the properties that pure sampling noise would?
No. You have something more, but that something more is negligible compared to the variation you have in biological, in two different biological samples. So if you're interested in the variation across communities, the true biological variation is much larger than the technical variation. So the patterns that you observe are driven by biological variation, not by technical errors.
I see. Okay, good. And I think that kind of gets to the last question too. But there's some more that you should check out in the comments.
Okay.
All right. So thank you very much. We should move on now. So next up, we have Maitreya Dunham from UW, talking about microbial diversity between and within species of beer. And she's from UW, but specifically from a boat right now, right?
That's right. Very Seattle, right?
Yeah. Okay, take it away, please.
Great. Thanks again for the invitation. This is a really fun session. And so I decided, in keeping with the diversity theme, I'd tell you a little bit about an older story that is partially published, and then also a newer project that we just put on bioRxiv like last month. And these are great collaborations, both with folks from my lab. The part one will be from Ivan Lyachko, who is a postdoc and has now started a company on the basis of this. So I want to make sure I declare that conflict of interest, and hopefully I won't sound like a shill. And then the second is led by a graduate student, Chris Large. And we've also been really lucky to have a number of excellent brewer collaborators. And so I'll try to acknowledge their contributions as we go. But it's been a really fun kind of citizen science initiative, and also working with professional microbiologists in addition to the scientists in the universities. Okay, so there are a couple different ways that beer is historically made, and we really think that it's going to be a great model system for understanding microbial communities and understanding domestication of microbes. And the two stories that I'll tell you today kind of highlight different aspects of what we think of this as being a good model for, and also understanding a real community doing real things, right? So the first is what was done, for eons before we started the modern brewery system, and that is doing open or quote unquote spontaneous fermentation, right? That was actually a big research topic for like Pasteur. Is this really spontaneous, or do these come from somewhere? And of course, these are microbes from the environment, which are able to colonize any sort of a nice food source, such as a bucket of nice boiled grain that's like sugar water, essentially. And so these are mixtures of microbes and not pure cultures. The modern brewery setup, after they figured out how to do pure cultures in Carlsberg Brewery, among others, in the 1800s, is that we now use or focus more on using specialized domesticated strains of brewing yeast, which are now commercially produced and which are clonal. And you'll see I put clonal in quotes here, because what I'm going to show in the second part of my talk is that they're not clonal at all. And there's some really great fun evolution going on in these fermenters in situ in breweries today. Okay, so let's start with the open fermentations and look at their communities. So this was a great collaboration with Cody Morris here, who's a brewer. At the time, he had a brewery called Epic Ales, And Epic Ales had been making this beer called Old Warehouse. And that's because their brewery, pictured here on the label, was literally in an old warehouse. And so Cody decided he's a very adventurous brewer, and he wanted to make a beer that kind of represented the place. And so he took buckets of wort, which is just that boiled grain mixture, and placed them all around the brewery, and then let nature take its course. And we got to go on a tour when we were taking the sample with him, and it is an old warehouse for sure. Like there was like a dirt floor in the basement, there's cobwebs, so the types of bugs living in this place we thought would be really fascinating. And so what he did was, after fermentation had completed in these buckets that he'd placed around, he went around and tasted all of them and took the one that he liked the best and used it to inoculate a wine barrel. And so by the time we went and visited Epic Ales, he had been keeping this going for about 3 years, where he would bottle half of it and then top it off with wort, and bottle half of it and top it off with wort. So this was a living community in here, which had been evolving and competing with each other for years. And when we arrived, he had just topped it off, and you could look in the bung here and see just roiling culture. It was amazing. You could tell it was alive. So we took a sample. This is Ivan's hand here. So we took it back to the lab and started to try to see if we could characterize what microbes were in the Old Warehouse beer. Before I tell you the results of that analysis, I want to mention the technology that we used to do that. And we started working on this because a graduate student in Jay Shanduri's lab, Josh Burton, was working on using Hi-C as a method of doing better genome assembly. And so we thought it might actually have a great application to doing better metagenomic assembly as well. And so, as has been described earlier today, the most typical way of doing metagenomics on a mixture of microbes is to grind them all up and sequence them, right? And so what's great is that you get all the different sequences coming from all the different bugs, but of course they're not beautifully color coded so that you can tell which genome bits came from which of these bugs. And of course, the field has come clever ways of making these matches and reassembling these genomes from these mixtures. The trick that we used is using proximity ligation in order to help us do this binning strategy. So the basis of this is a method called Hi-C, where you essentially just treat your DNA with a crosslinker. You fragment it, you use like half the NEV catalog to end up with this at the end of the day, where you can sequence these junctions where you get a little bit of this piece and a little bit of that piece. And so what that tells you is that these two pieces of DNA must have been close enough in three-dimensional space to be able to be connected by this chemical crosslinker. So Ivan and Josh realized that that doesn't just tell you that they were close to each other, it tells you that they must have been in the same cell, right? So I still remember Ivan coming into my office with this idea, and I was like, you know what? I don't know anything about metagenomics, but that is such a cool idea, and it's so going to work that we need to do it. And so we started working on it, and it's worked out really well. And like I said, Ivan has gone on to start a company on the basis of it. And I should mention, it was such a good idea that 2 other groups were also working on it at the same time. And so it's really interesting to see how the different groups have kind of gone on to implement things slightly differently and also use it for different applications. So I'd encourage you to go and look up their bodies of work. Okay, so what this does is it now allows us to, instead of having this big mixture here, it allows us to use these crosslinks to cluster reads, essentially. So what we do is we create little contigs, and then we bin them according to who's got links between them. So this lets us now sort everything out. And so here's the cartoon version, but what about real data? So what we did was we went into my freezer and we got out all of the yeast strains that had been sequenced, and we made a synthetic community out of things that would never see each other in real life, but that we knew who they were. And we assembled the community and then sequenced it all together like this. And so what you're seeing here is a visualization of the clusters that are created by Hi-C. And then the other nice feature of this is, because Josh had been working on Hi-C for better genome assembly, we're also able to assemble nice little genomes from this as well. And so if we go and look, these are all matching these clusters, the things that we know that we put in there, which was really great to see that it seems to have worked. These gray lines represent a few different types of things in the data. One is that there are sequences that are very closely sequence similar among the different species, and so it could be representing that. And in fact, in other work, we've been able to show that we and others have been able to show that you can see things like a plasmid that's kept in multiple different species if you look at these more carefully. And some of them are just artifacts of the mapping. Okay, we also went next door to the microbiology department and got some bacteria and archaea to make a more kind of diverse population, and we were able to find those as well. Okay, so now back to the beer. So what did we find in the beer? So one reason we thought that this would be a good sample as a test bed for our method is that beer communities have already been characterized to some degree, and they're relatively simple. There's not a ton of species going on, and so we thought it'd be a good place to start. And so sure enough, what we found was only a small number of species, and many of them were the usual suspects. So I'm a yeast geneticist, so of course I was thrilled to see my favorite Saccharomyces cerevisiae, the standard brewing yeast. But we also saw a few other species that are typically found in these mixed fermentations, including Deckera here, which you might have heard called Brett, for it's another yeast. And it offers that kind of a funky flavor to a lot of these open fermented beers. It's often described as wet dog or horse blanket. As you can expect, a little bit of that goes a long way, but it does add to the complexity of the flavor. There are also some lactobacillus, which is expected and adds that kind of sour beer character. And we also found some acetobacter, which creates acetic acid, and so is a little less favored in a mixture like this, but is still an expected component. What we were really excited to see was this cluster here. And this was a yeast that we weren't sure what it was. And we initially got really confused by it because when we tried to do the mapping of the reads here, only half of them mapped to the database. And we were like, what is going on here? It's either something really diverse or something else is going on. And so the nice thing about working with beer is that essentially, sugar water is the same as yeast media in the lab. So you can actually cultivate many of these species individually and grow them up and sequence them as clones. And so we were able to clone purify this bugger here. And here it is under the microscope. It makes these really neat little palisades. And half of the genome comes from Pichia membranifasciens, which has been associated with fermentations previously, but half of the genome was actually from something else. And we still don't know what this other Pichia is. We've never been able to find the pure parent of the other species here. So this is an interspecific hybrid, which is in this beer. And we named it Pichia apotheca just to have something to call it. And I've now sent it to hundreds of home brewers and some professional brewers who are starting to send me weird beers back in the mail too. So it's been a really fun project of giving people a new species to play around with. It's not the world's best fermenter, but it does have some interesting behavior and flavor components. Okay, so what was so neat about it being a hybrid? We actually in my lab have a whole set of projects on hybrid evolution, so I was really excited to find this. But what was cool was that we could figure this out from the Hi-C data. So while a normal shotgun sequencing, you can't tell the difference between two species that are separate versus two species that have created a hybrid. Using the Hi-C, you can. Because if the two species were just individuals, they would only create links among their own genome. But if it's a hybrid, you get links between the genomes. And so that lets you cluster those sequences in a way that lets you tell they were in the same cell. Okay, so that's all published. You can go read more about it in the papers, but I just wanted to say that we're still using this method to do more fermentation metagenomics. And one exciting thing is we've gotten a time course of some Belgian fermentations, which have been pretty well studied by other means in terms of the succession of species. But one thing that we found that was novel in this is another hybrid. So this is a Cerevisiae Kudryavzevii hybrid, so two Saccharomyces species that have created a de novo hybrid. And Barbara Dunn, a visiting scientist in my lab, has been working on this and trying to understand more about the complexity in these Belgian samples. We've also collected samples from ciders, wines, more beers, and we're always on the lookout for additional interesting things. And your sample could be here. I've really enjoyed getting introduced to all the various brewers who are using all these creative techniques in their breweries. And they're often really eager to collaborate, and I've learned a lot from them. Okay, so one downside of this project is that we went and we took one sample of the beer and we have a snapshot. And unfortunately, Cody's brewery went out of business and Epic Ales is no more. And the old warehouse barrel got decommissioned. Don't worry, he's fine. He started a new brewery. He's now brewing under the name Mollusk, which is also quite interesting and good. But it would be nice if we had, time courses like was shown earlier. We could follow both how these species are evolving by mutation, but also by population abundance over time. So I think that sounds pretty hard. We'd like to do it one day, and we're getting to some of it through the Belgian time course. But the next project I'm going to talk about is just using one species as a simpler way to get at that. And so this is a project that we just posted to bioRxiv. And it's looking at just passaging Cerevisiae in brewing and seeing what genome changes happen as you reuse Cerevisiae. So, as opposed to this kind of boutique method where you're inoculating a wine barrel and kind of treating it in this interesting way, the more typical way that microbreweries work today is they purchase yeast from a propagation company, and they'll inoculate a fermenter with it. And they'll let that fermentation run. Yeast, when it's exhausted, will clump together and sink to the bottom. So what's easy to do is to collect the yeast after the fermentation is over, and then you can just pitch it into the next fermentation cycle. And so this is called repitching. And typically the usage rate is about 5 to 10 cycles before you throw it away and go back to your yeast company and get more yeast. And so why is there this 5 to 10 cycles? It's in part because there are changes to how the yeast behave and how much sugar they eat and how much they clump together, and also the features of the beer, how it tastes and smells. And the causes of these changes are, there's a few things, right? Contamination is something you always have to worry about. There might be metabolic changes as the yeast adapt to their new environment, and there are also nutritional deficiencies in wort. So it's low in a few things like zinc, for example, and sometimes people add that back to try to boost their yeast. But it could also be mutations and evolution, right? And I came across this great figure on a brewing website, which is what happens if you reuse your yeast too much. You eventually make zombie yeast or something. And so nobody wants zombie yeast, right? So except Tom Schmidlin, the head brewer at Postdoc Brewing. So he was the first person who I started this collaboration with, and we've now expanded to a few more breweries, which I'll tell you about. But when I met Tom, I was astonished to learn that he reuses his yeast for 40 or 50 brews instead of the 5 to 10 that's much more typical. And he does this in part because purchasing yeast is an expense, a non-trivial one. And like I said, breweries go out of business, right? They're operating on really thin margins. So this is helpful for him to be able to reuse his yeast. Also, I have a theory that Tom's got a little bit better sterile technique than some places just because of his background as a scientist, as you might guess. Okay, so Tom agreed to take samples for us every time he brewed beer, starting from buying the yeast to the end of when he decided to quit. And his decision to quit is based on his own perception of how things are going. Is the yeast performing well? Is viability staying high? Does it taste the way he wants it to, etc.? So this first experiment I'm going to walk you through is 26 repitches. And in order to introduce you to the species inside, I want to just mention that it is not what you're thinking. So if you're familiar with yeast, you might be thinking of the lab yeast, where we've got 16 chromosomes. If you're a diploid, you've got 2 copies of each. You're usually nice and homozygous, well-behaved. Brewing strains are not that. So brewing strains are probably from historical hybridization, an intraspecific hybridization of a sake yeast probably and a wine yeast. And so it's essentially a tetraploid with 4 different parents, because both of those parent species or strains were really heterozygous. So on top of that, it's had massive chromosome gain and loss events and recombination events over the course of its getting passed around from brewer to brewer and then getting propagated in the breweries. And so this is just the one strain that I'm going to talk about today, but of course there are hundreds of brewing strains with different histories that people use. But this one's an American ale strain that is often called the Chico strain, based on the Sierra Nevada brewery who popularized it. And it's just got a crazy messed up genome. It's really been fun to work on, but has made our life a little more tricky trying to figure things out. Okay, so what happened after these repitches? Well, first of all, the chromosomes changed more. So we ended up with an extra copy of chromosome 5 here. So we started out with 3 copies, which is lower than the average ploidy in this strain. And now it's kind of almost reverted back to 4 copies. And so you can see the data here. This is just using read depth as a measurement of copy number. And so these are different time points, and this is an estimate of the frequency. And so by the last time point, this actually had risen to about 40% frequency in the population, which I was truly astonished by, given that the population size in these fermenters is something like 10 to the 14th cells. So you're doing pretty good to make it to 40% of a population size that large. The other types of changes that we saw were more subtle in that they didn't change copy number, but they changed the allele balance. And so here's a cartoon of chromosome 8 in the starting strain. Chromosome 8 maintains all 4 of the initial haplotypes on one arm, but the other arm has actually already undergone mitotic recombination, and we're down to 2 haplotypes, and they have a minor one and a major one. After the repitching, what we saw was instead 2 of the minor haplotype and 2 of the major ones, so it balanced it out. And the data to understand this is really complicated. It's an allele frequency plot. So you can imagine, like, if you're homozygous, then you only have choices. If you're looking at allele frequency at different sections of the chromosome, right, you have to be fixed if you're homozygous. If you're heterozygous, now you can have some sites that are at 50% allele frequency. And that actually is the same if you're a diploid with 1 copy of each, or if you're a tetraploid with 2 copies of each. But things start to get more complicated once you throw in mix and match here, or different copy numbers. And of course, if you have multiple haplotypes to keep track of, you can start getting all sorts of patterns based on the variants that are shared among these haplotypes and which ones might be unique to a single haplotype. So here's what that data looked like for chromosome 8 from the brewing time course. And what you can see here are all the different haplotypes on this arm, but this arm has just the 2 that are 3 to 1. And over time, what we saw is this kind of shift at the end of the chromosome to be more of a 50-50 mix. And this pattern where it's kind of a gradual shift, we initially didn't understand that until we started sequencing clones. And so what we found was that what this represents is actually a large population of independent recombination events, which are sweeping together as a cohort. So it's kind of massive clonal interference, right? So the dotted lines here represent the breakpoints on each of these clones that we sequenced, which each had a different location. So that also allowed us to really zoom in on this end here, which was the smallest recombination spot that we saw. And we've got some good candidates in here that I won't have time to talk about today. Okay, so does it replicate? So after doing this, which took like 6 months of beer and then all the analysis, Tom did it again for us, collected a second time course. And we were also able to find brewers at Elysian, which is a local Seattle brewery, Drake's and Red Circle, who use similar strain backgrounds, similar but not identical. This is one family of strains again, and carried out time courses for us and collected samples. And to our great astonishment, we found not just the same types of events, but exactly the same chromosomal events. So we saw this chromosome 5 copy number change and this chromosome 8 recombination. And one thing that was really interesting was when we started looking more at these ancestral strains, some of them actually already had these mutations which were accumulating de novo in our fermenters. So what we think that indicates is that what we're able to capture over these shorter term serial passaging experiments, which, to my mind, look a ton like experimental evolution, right, is actually indicative of patterns that you see over longer term strain evolution as well. And so here's just a tree. We started collecting all the Chico strains we could get our hands on. Chris posted in some brewing message boards and was able to get some samples of people's house strain, or a bunch of different companies sell strains that have different names but are all derived from the same ancestor and things like that. So I didn't fill in all the names here, but the full tree is available in the preprint if you have some favorite strains. But what was interesting is that, this strain set, which is kind of the outgroup here, so BRY96 is pretty close to the true ancestor of this group here. It starts out with 4 copies of chromosome 5, but then this whole section here has lost chromosome 5, 1 copy, but now some of those evolution experiments have gained it back, right? And another example here is that chromosome 8 mitotic recombination, where it has happened at the root of this part of the tree. So all of these strains already have it. And then it happens de novo in our evolutions on this side of the tree. So we've been really excited to try to figure some of these out. There's one more mitotic recombination that we saw multiple times on chromosome 15 once we added in all these new replicates. And it's really exciting to think about this in the context of, what has happened to these strains historically. They clearly have way too many mutations, and you can't do genetics on them because they're tetraploids that won't go through meiosis. But can we use de novo mutation and kind of experimental evolution to understand how these mutations behave and what they might be doing over the course of a strain evolution like this? And also I want to mention, of course, this is just a little tiny part of the beer diversity tree that's focused on the American ale strains, but there's of course a bunch of strains out there all with different characteristics and things. Okay, so in the interest of time, I'm not going to go through all of the results in the paper, but I just want to send you to the preprint, and I'd love to hear comments on it if any of you read it. We found one candidate mutation that we think might be driving the recombination event, and that's a loss of function mutation in BAT1, which is involved in branched amino acid biosynthesis. And Chris and I just happened to be at a meeting with one of the world experts on BAT1, and they were interested in the mutation and ended up writing an entire paper about the interesting features of this mutation. So that just came out like literally a week ago. So please go take a look at that too if you're interested. And the paper also has more information in it, like what do these mutations do? Why are they having such success in our fermenters? And how does it make the beer taste? So one fun thing that we did was we brewed beer with the ancestor in 2 of the evolved strains. And Tom brewed beer such that we could bring it to a home brewing convention. And we had all of the audience do a sensory analysis, which was great. And we're able to collect tasting data from semi-experts on this. And so we were able to detect differences in taste that also the mass spec detects when you run it through to measure those things. We have more phylogenetics analysis. And like I mentioned, these genomes are crazy. And it became very clear that we needed to have a better genome assembly and better phasing. So we've been working with Joseph Schuscher's lab to use nanopore sequencing to get more information, and particularly the telomeres of these strains all mix and match and are a mess. So the genome assembly with nanopores really helped straighten out some of that. Okay, and also we're doing some other things. Even though I focused on the reproducible mutations that we see, there are other mutations in some of these clones that make the comparison of phenotypes not truly isogenic, right? So if we have the recombination plus a few point mutations, it's hard to tell what's what. So we're now using CRISPR to make these recombination events and try to and also point mutations and try to reconstruct truly isogenic strain pairs that we can do better comparisons with. And finally, we're on the lookout for more breweries doing this sort of thing that would be willing to collect strains for us. Time courses and also interesting practices. Like one brewery that I'm working with now is doing top cropping, where they collect the yeast from the top of the fermenter instead of the bottom. And so we're interested to see if that makes a difference in how these things evolve. Okay, and with that, I'll end. I again want to thank Ivan and Katie, who worked on the first part. And then here's Chris. Actually, Chris is wearing his postdoc brewing shirt here, which is nice, for the second part. And both Chris and Noah actually are home brewers themselves. So it's been kind of a fun project. This started as an outreach project on a broader impacts for an NSF grant, and it's turned into so much more. And so it's just been really great fun to work on and to get to work with all these great brewers. And so I'll take questions now. Thank you.
Great, super fun. Okay, we don't have time for all the questions, but I'll just start asking them in rough chronological order. So Eric asks, yeah, so what do you think the kind of key properties under selection are?
Yeah, so we've tested this a little bit. One is just simple growth. So we came up with a lab recipe for making truly sterile wort. Of course, in the brewery, everything is just sanitized, right? So we had to reconfigure a few of our protocols to work better in the lab. And we do see a growth advantage for the strains that carry some of these mutations. So we think that that's the most obvious thing. But we also have some preliminary data showing increased ethanol resistance of some of them. And many of the breweries that we work with make an awful lot of IPAs, right? I'm in the Pacific Northwest here. It's what we do. And so, you know, some of these things go to like 8% alcohol. And so there could be some interesting modern selection pressure for these increased alcohol concentrations and increased alcohol tolerance. That's why these adaptations have not pre-existed in these strains before now. The other interesting thing is the population genetics of this. And you think about how these strains get passed around, and they're going through probably single cell bottlenecks as they're getting passed from brewery to brewery and from yeast propagation company. And so there's a possibility of fixing bad mutations through this process, but then they have a chance to revert once they're put in the super competitive environment of the fermenter. So we think that that's another thing. They're not beneficial. They're in fact fixing a deleterious mutation.
Well, that gets to the next question from Dan Falush about bottlenecks. So yes, you're saying, okay, they're often bottlenecks, and just I'll try and paraphrase his thing. Did it look like the strains were bottlenecked when they first arrived at the brewery? So it's all essentially clonal. And was there any sign of maintenance of polymorphism during the repitching process?
Good question. So, some of these companies have internal yeast propagation centers, but most of them buy the yeast from an external site. And so what the propagation company does is they have a stock and they grow it up. And then sometimes they'll grow up one big batch and subdivide it to send out to different breweries. So we actually don't have perfect information about how many generations things go through before they hit the brewery. But in the second time course from postdoc brewing, one thing we noticed was that the time 0 already had about 10% of that chromosome 5 copy number change. So some of these mutations are already being selected for at the propagation company step. And then the inoculation, they get them off to a head start and then it takes over. That particular time course, that chromosome copy number change went to a much higher frequency actually than in the other experiment. In terms of once it hits the brewery, there aren't big bottlenecks in the brewery. So they're using about 1/3 of the yeast volume to repitch to the next population. So that's a ton of yeast. Although upon interrogation, we did learn some things like, if they're not going to use it immediately, they put it in a keg and throw it in the cold room for a little while. And so there could be some interesting selection pressures at work for some of these handling steps.
Even a Tijovic asks, do you have a sense of how much cell death division there is? It's like how many generations, how strong does selection need to be to produce the observed frequency changes?
Yeah, good question, and something I forgot to mention. So it's about 3 generations per repitch. So if you think about what the brewer wants, they want the yeast to convert the carbon to ethanol. They don't want to convert it to more yeast, right? So they're trying to repitch actually at a rate such that there's not a ton of cell division, but there's more fermentation. And so there are only about 3 generations per cycle. We have estimated the selection coefficients on some of these based where we have nice time courses. And we think that the collective selection coefficient of the cohort sweeping is on the order of like 3 to 6%. So that's non-trivial for sure. But even pretty subtle things can happen. And the mutation rates are, of course, is the other point of this. And so that I didn't give you the data for this, but that chromosome 5 copy number change also happens multiple times independently within the population and kind of sweeps as a cohort. So we're seeing things happen many times within the same population and also across populations.
Great. Okay, so there are more questions, so you should check out the chat, and we should move on to our final talk. So Nandita, can you come ahead and share your screen? And last up, we have Nandita Garud from UCLA talking about evolution in the human gut microbiome. Okay, I see a message that you've started screen sharing. And still.
Can you see my
blank.
screen?
We can only see a black message. At least I can only see a black message. If someone else on the panel can see something, please weigh in. We see you, but yeah, I guess you stopped screen sharing.
All right, let me try it again. Can you see this now?
Nope. So somehow between, an hour ago when we checked it, or 2 hours ago when we checked it, and now.
Maybe only start the presentation after you shared.
Okay, let me try that.
No.
Nope, still black.
Okay, maybe I'll rejoin the Zoom.
Okay. In the meantime, Maitreya, if you want to come back on and talk about a little bit more about high C, you can see there's some questions about that.
Yeah, happy to.
Thanks. Yeah, so you see there's quantified false positive rate for the high C metagenomics and quantitative insights on the limits of sequence diversity for communities where HiC can still be useful?
Yeah, those are both good questions. And really, I showed like the very first proof of concept experiment. And we've now had a chance to collaborate with a bunch of other groups. And of course, Ivan's company has done a bunch of work with more complex communities. And you can actually do pretty well. We've done some medical samples and some human microbiome samples. And you do have to sequence pretty deep though. So you end up kind of having a sensitivity related to how much you sequence. So I'll answer the rest of the questions in the chat since we can now see your slides. Great.
Awesome, thank you.
Great.
All right, go ahead, Nandita.
I'm relieved. All right. Well, it's a really great pleasure to be able to share my work with you today. And I'm going to be speaking about evolution in the microbiome, or as I've titled this slide, the bacteria evolving within us. So we've learned a lot about evolution in the lab. For example, famous experiments by Lenski and colleagues have shown that E. coli grown in test tubes in the lab can evolve rapidly and repeatedly. But this picture of evolution comes from one of a simple community in which you just have one species being cultured in a test tube. And the question is, how does this picture of rapid evolution translate to that of the human gut microbiome, where you have an amalgam of bacteria, archaea, fungi, and viruses all interacting with one another? And moreover, you have the strains within each species inside the human gut. So it's actually a very complex community compared to the ones that we see in the lab. And instead of responding to a selective pressure via a genetic adaptation, you might actually see species fluctuations over time in response to that change in pressure. Alternatively, you might get an invasion of a better fit strain from the outside environment. Or you might actually see evolution in which you have a genetic change that propagates through its subpopulation. So when I talk about evolution, I just want to be really concrete and say I'm talking about a change in allele frequency within a population rather than a change in species composition. And there's actually an abundant opportunity for adaptation in the microbiome. It's been estimated that that almost half of our cells are microbial, and that combined with generation times, short generation times, and mutation rates, it's been estimated that we have almost 1 billion mutations entering our microbiomes daily. So why should we care about this process of evolution inside of us? Well, so first, we want to know if this even happens. To date, we have only really witnessed species fluctuations and really quantify that extensively. And we really want to understand if there's more to it than just species fluctuations. But additionally, the genetic variation inside of us actually confers important traits not only to the microbes, but also to human hosts. Genetic variants in the microbiome have been associated with the ability to digest food. It can confer antibiotic resistance and even change the way our bodies respond to drugs. The processes that give rise to these genetic variants are evolutionary processes of drift, mutation, migration, adaptation, and recombination. And yet we have so little understanding of how these processes occur inside of us in our complex communities. And so today I'm going to be speaking about the role of evolution inside of us, and specifically in commensal microbes in healthy hosts, where there's no obvious selective pressure like an antibiotic or a change in diet. And I wanted to acknowledge my co-first author on this work. It was recently published in PLOS Biology, and Benjamin Good was a really fantastic collaborator on this. Okay, so the overview of my talk is that first I will share with you how we track evolution within the microbiome. Then once we figure out our methodology, we'll talk about evolution on short timescales, then over our lifetimes, and then I'll share with you a little bit of new work that I'm pursuing with a collaborator, Jesse Shapiro, and his student, Naima Mahdi, on evolutionary dynamics in complex communities. Okay, so traditionally, a lot of 16S data has been collected for the microbiome, in which we can barcode which species have been present. But this actually doesn't really inform us about the genetic variation in the rest of the genome. And so therefore, we can't actually witness evolution with these types of data. And instead, what we could use is whole genome shotgun sequencing, which allows us to sample the rest of the genome in a pretty high throughput manner. It's not perfect data because these are very short fragments of only 100 base pairs. And you end up with this really complex mixture coming from many species. But at the same time, though, it actually allows us to see processes of evolution and invasion. And so that's the kind of data that we're going to be using for this project. And specifically, it's from the Human Microbiome Project. Now, how do we detect evolution with shotgun reads? So we can see what we want to hope to see is mutations that rise from low to high frequency with time. But this is really complex. It's like as if we're looking for a needle in a haystack. And first, we need to simplify this data and perhaps look at genetic variation within individual species one at a time. And so what we can do is we can map reads to reference genomes, as I'm showing here. And so we can align them and then identify mutations inside of each species, such as this one at 50%, that correspond to a polymorphism inside of a host, inside of a host species pair. So ideally, to quantify evolution, once we map those reads, we'll then identify allele frequency changes from low to high. But this is actually harder than it looks. A complication is that there could be multiple strains colonizing a host at high frequency, and the fluctuation of those strains could generate signatures of allele frequency changes that can be confused with evolution. So let me expand on this point a little bit further because it's fairly important. Basically, what we're talking about when we talk about evolution is a handful of changes that may accumulate over short timescales of about 6 months. When I'm talking about invasions, I'm talking about strains that may come from the broader population that have had a lot of evolutionary time to accumulate a lot of mutations. And in fact, the average distance between strains collected from different individuals is about 1%, which is actually quite a lot of differences compared to what you'd expect over a short time scale. So we need to figure out how to resolve the lineage structure inside of hosts in order to be able to distinguish evolution from invasions. Otherwise, our signal for evolution will be swamped out. And the tricky bit is that all we're witnessing actually are allele frequencies, and we have to somehow infer that latent space of strains. So what I'm doing now is I'm actually going to plot the distribution of allele frequencies for different hosts for the common bacterial species, Bacteroides vulgatus. And what I'm plotting on the x-axis are allele frequencies from 0 to 50%. And on the y-axis are the fraction of sites with that allele frequency. And what we see right off the bat is that there's a lot of variation across hosts. You can see that there are these modes that occur at different frequencies. So what is actually generating these figures? So let's take a look at this very first person. And you see that there's a mode at 15%. And it turns out that this host is colonized by two prominent strains at 15 and 85% frequencies, respectively. And these strains have had a lot of time to diverge before colonizing the host. And so the mutations that show up at 15% are actually mutations that have accumulated long ago on the background of these different lineages. And so this mode actually is representative of the relative abundances of these strains inside of hosts. And you can see similar patterns here, where this host was colonized by 2 strains at roughly 50-50 frequency, and this host seems to have been colonized by some complex mixture of 3, 4, or some other number of strains. Now, our goal here is to identify the genotypes corresponding to us strains that we can actually track evolution on the background of that strain. And so what we want to do is focus on those samples in which we can confidently phase at least one lineage as genotype. This sample happens to be very complex, so we'll ignore it. This sample, though, is very easy to phase because you can actually assign the common allele to the common strain, as shown in the red reads and the red lineage here. But this sample happens to be more complicated because you can easily confuse the site that belongs to the red versus blue strain. And so we need to basically identify those samples that have a clear dominant lineage so that we can assign mutation to that or genotype to that lineage, and then identify mutations on top of that lineage with time. And so this is the kind of signature that we're looking for and these kinds of samples that we're using. And you might be wondering, how many such samples do we have? We call them quasi-phasable samples. And colored in dark blue for the top 40-ish most prevalent species, we see actually that we have about 801 species host pairs that we actually can work with. And so this is actually a tremendous amount of data and allows us to do a lot of PopGen simultaneously across many species. And so going forward, actually, we're going to combine the data across species so that we can have better power to say something more generic about evolution in the microbiome. So now that we've done this hard work of trying to deconstruct the lineage structure inside of hosts, we can look at evolutionary dynamics on short timescales within hosts. And so again, the signature we're looking for are these mutations that rise to high frequency on around 6-month timescales. And what I'm going to plot here are the fraction of comparisons that have N or more SNP changes. On the x-axis are the number of SNP changes. And this is what we get for our roughly 800 data points. It's this kind of distribution that has a mode here and a mode here. And let me walk you through it. So this is a plot plotted on log scale. And so therefore 88% of our populations actually have 0 SNP changes. What's new though is that 9% have actually a handful of SNP changes on the order of 5 or 10. And then another small handful have around 10,000 SNP changes. So this vast difference in magnitude of number of SNP changes, 5 to 10 versus 10,000, suggests that there might be 2 processes driving these SNP changes inside of hosts. What could they be? Well, going back to our original outline, it actually could be a mixture of invasions and evolution. Remember that in evolution should occur on short timescales and should pick up a handful of SNP changes, whereas invasions are expected to bring in a large number of SNP changes due to there having a lot of time passed between the lineages that are coming in and being replaced. And in fact, if you look at the number of SNP changes within hosts, we actually see on average a few hundred within hosts versus 10,000 between hosts. So going back to our plot, we can now put on top of this plot another line where we're looking at the average number of SNP differences between pairs of hosts. And you can see that we, again, have on average about 10,000 SNP changes between hosts, allowing us to distinguish within host changes as either evolution or invasions. So this was actually really exciting because it suggested that evolution is a phenomenon that we can actually observe in healthy hosts in our commensal microbes. And it moreover is occurring on really short timescales of just 6 months. this means that potentially the genetic changes in our microbiomes could potentially help us to respond to selective pressures on short timescales. Now, we wanted to ask further, are sweeps seeded by de novo mutations or recombination events? By de novo, I mean a brand new mutation that spontaneously arises. By recombination, I mean something that was taken up from the broader environment, such as in this cartoon, where you have a donor giving a gene to the recipient, and then you have a swap out of genes and exchange of genetic material. So to answer this question, we took the mutations in this part of the plot that are changing due to evolution, not invasions. And what we did was we plotted whether sweeps are the number of SNP changes versus the derived allele prevalence across hosts in black. And we compared it with a de novo expectation in gray, in which we just kind of permuted the data. And you can see that there's actually an excess of high prevalence SNPs changing within hosts, suggesting that these mutations were already pre-existing in the broader population. Now you might be wondering, well, could this be to recurrent mutation potentially? And answer is unlikely, because we actually see depressed DNDS values at these intermediate prevalences across hosts. So these two pieces of evidence suggest that the mutations have been lingering for some time in the population and for sufficient time for purifying selection to purge out deleterious variants, resulting in depressed DNDS. So this suggested that recombination may be playing a role, but moreover, we saw a lot of gene gains and gene losses, and gains losses can be expected via spontaneous de novo mutation, where things drop out, but gains have to happen via uptake from the broader community. And indeed, we see actually a roughly equal distribution number of gains and losses. So this suggests again that recombination plays a role inside of hosts in seeding sweeps. So now we wanted to ask, how do the evolutionary dynamics look like over our lifetimes? Specifically, do the microbes inside of us keep adapting such that it becomes difficult to replace the resident lineage that has become so well adapted, or are there limits to adaptation? So remember that we saw a small fraction of populations becoming replaced in just 6-month timescales. Question is, after 20 years, does every strain get replaced after 20 years? Or is it that only some hosts are extra susceptible because they are taking antibiotics or traveling? Or is it that strains become so well adapted that replacements become less likely with age? And ideally, to answer this question, we would sample a single host over a few decades, but obviously this is very difficult to do. And so what we can do instead is to leverage adult twins to test the long-term dynamics. So a paper by Corpella et al. Showed that cohabiting teenage twins and siblings share many strains. And it turned out that there was a nice data set by Shi et al. In which there were 125 adult twin pairs that grew up in the same household, after age 20 or so, moved out, and then lived apart for 40 years. And we asked whether the adult twins shared the same strains. If so, then we could see evidence for potentially long-term evolution. But if not, it suggests that everything gets replaced over time. And so what we did was we then added a line to this plot, looking at the number of SNP differences between twin pairs, and here it is. It almost perfectly overlaps what's going on between hosts. And so this suggests that actually the strains within hosts over time are replaced over our lifetimes. So on short time scales, evolution seems to occur, but on long time scales, replacements seem to dominate. Why these replacements occur is somewhat of a mystery. Maybe it's occurring in bursts due to antibiotics. Maybe these replacements are occurring gradually over our lifetimes, but these are questions that are yet to be resolved. Okay, So this brings me to the very last portion of my talk, which is on evolutionary dynamics in complex communities. And this is a new project that I'm taking forward with Jessie Shapiro, Daisy Chen in my lab, Naima Mahdi in Jessie's lab, and Richard Wolf in my lab. And the question we are asking is whether species diversity begets genetic diversity in the microbiome. This is a paper that is a follow-on from Naima's paper that's right now on bioRxiv, where she's asking about does species diversity beget species diversity in the microbiome using 16S data. And now here what we want to ask is whether the species complexity actually results in more or less adaptations, or even just asking more simply whether the species complexity results in more polymorphism within a focal species. So here what I'm drawing is an adaptation from Naima's paper where you have two species, and within a focal species, you have some diversity. And the idea behind DBD is that as the community complexity gets greater, new niches arise, giving rise to the opportunity for new adaptations. And we can plot that relationship here where the community complexity increases along the x-axis and evolutionary modifications increase along the y-axis, reflective of new opportunities to diversify. And here is a contrasting hypothesis in which when you have more complexity in the community, you actually constrain opportunities for diversification because niches become filled. So here you would actually see in this ecological constraint hypothesis, inverse relationship where the more complex your community, the fewer adaptations you should see. And so the question is, which of these hypotheses holds true in the human microbiome? And Naima saw that for human gut microbiomes, the DBD hypothesis seems to be generally true at the species level. But now let's take a look at the within species level. And so first, what we're actually going to plot is the genetic diversity per species per host. So it's roughly plotting pi, or amount of polymorphism for different species, as a function of community complexity, which is measured here as Shannon diversity. And we can see a positive trend in which we see a positive trend in where you have more polymorphism within species for more complex communities. And breaking this down by focal species, here are two examples in which we have a really strikingly positive trend, whereas some other species have almost zero, or maybe you would even say negative trend. So this is already some evidence that DBD seems to be occurring for at least some of the species in the gut at the polymorphism level. Now we can ask about whether adaptations seem to increase in more complex communities. We can take the mutations that are rising to high frequency due to evolution inside of hosts and plot the number of evolutionary modifications versus community complexity measured by Shannon diversity. And this is broken down by species, and you can see that roughly we do actually see a positive trend with Shannon diversity. We can even take a look at the number of gene gains and gene losses. And again, we see gene gains with increasing community complexity. This makes sense, more material to draw upon, but the gene losses was actually something very exciting because it wasn't to be expected that you'd see a lot of gene losses in a complex community. And this to me actually suggested potentially some evidence for the Black Queen hypothesis, whereby public goods are provided in more complex communities, thereby eliminating the need for a focal species to make its own resources to support itself. All of this work is in progress and something I was really excited to share with you today, but we're still putting the finishing touches on this. So I wanted to just conclude and say that hosts are oligocolonized by a handful of strains per species. On 6-month timescales, bacteria can evolve in the human microbiome. Replacements are rare. Over our lifetimes, bacteria are replaced. This suggests limits as to how well adapted bacteria become to us. The broader community of bacteria play a critical role in providing the material for recombination-seeded sweeps. Species diversity begets genetic diversity in the microbiome. I want to thank Ben Good, Katie Pollard, and Oscar Halachek, my co-authors on the microbiome evolution project I presented, published now in PLOS Bio, as well as Jesse Shapiro, Daisy Chen, Naima Mahdi, and Richard Wolf for the Species Diversity Begets Genetics Diversity project. And my lab is recruiting. This is us at UCLA, and we have a lot of fun together, even on Zoom. So thank you very much for listening.
Thank you, Nandita. First question from Mohan Ghaffari is about the DNDS measurements that you mentioned. So he's wondering what kind of timescales would those allele frequencies correspond to, and yeah, how strong would that selection need to be?
Yeah, so we plotted DNDS as a function of DS. I haven't shown that plot here, but basically we were able to look at patterns of DNDS as a function of how diverged these lineages are. And this actually follows really nicely with what Erik spoke about earlier, in which we see a really large range of divergences across bacteria in the microbiome. And that large range actually allowed us to see purifying selection in action, whereby really closely related strains had very high DNDS, well, high as in relatively about 1, whereas strains that were diverged on the order of 10 to the minus 2 had DNDS values on the order of around 0.1. So this inverse relationship allowed us to fit a very, very simple model in which roughly 90% of the genome is subjected to around a selection coefficient of 10 to minus 3. And obviously it's a simple model, but strikingly it actually kind of fit the most prevalent species in the human microbiome. So in terms of number of years, I suppose you'd have to just calculate given divergence of 10 to minus 2 and think about how long these strains have been lingering in the population, but hopefully that these numbers should give you a sense.
Good. So Erik asks, okay, so your observation could be summarized as saying that more diverse communities are more evolvable.
Potentially. This is fresh off the press, but yes, we see positive trends in which we see evidence for more evolutionary changes in more complex communities, suggesting that there might be more niches available.
Luis Rodriguez and Alexander Christoph asked some questions that I was wondering about. So, I mean, is there any argument, though, I mean, that you really see about which way causality is going here in this relationship between diversity within species and between species? And Christoph specifically asks about possible issues with technical artifacts where you're accidentally assigning reads from one species to another species.
Sure. So we have been actually very, very careful with regards to these technical artifacts. We care a lot about whether a read belongs to the focal species that we're looking at versus not. And so we implemented a number of bioinformatics controls in addition to implementing a model for sampling error when looking at evolution. So some of the bioinformatics controls include some of your standard thresholds for read mapping, genome coverage, and so on. Specifically, we were careful to exclude regions of the genome which had abnormally high or low copy number variation, potentially suggesting something funny about the reads that are recruited to that part of the genome. We made sure to actually exclude any region that showed any abnormal coverage across any sample in our data set, actually, irrespective of whether that abnormality showed up in a particular focal data sample. We basically left no room for ambiguity. We removed any read that could potentially map to more than one species. So we looked for any orthologous regions and removed those as well. So our gene blacklist actually was quite extensive. Thus, the conclusions that we make here are really conservative and in fact probably hide the extent of adaptation inside of the microbiome because we didn't want to be riddled with any errors. The distributions of genes, gains and losses, and also SNP changes don't suggest that there's anything erroneous creeping in because these actually look to be asymmetric, specifically with respect to the gene gains and losses. Had there been a read recruitment issue there, we might have actually seen more symmetry. So many pieces of evidence that we considered point to the fact that we've actually accounted for a lot of error here, not to mention the sampling error.
All right, so we're out of time, but just to that first part of the question though, is there any reason to think that the error of causation is going one way or another, or it's not both caused by some third factor?
So let me see if I understand the question. Is causation of what to what?
Oh, so you see a positive association between within species diversity and ecological diversity, right?
Oh, okay.
Yeah. It doesn't. What
Fair
about
enough.
causation?
Yeah, it's to be determined, but we are actually breaking this up one species at a time. And so we see this positive trend across individual species, suggesting that, I think it would be a little difficult for individual species polymorphisms to all independently contribute to more species complexity. It seems easier the other way around, where you have one variable causing, that being species complexity causing within species diversity.
I don't know. I eat a lot, that might increase species diversity and within species diversity because everybody's got more food. I don't know.
Well, that's a great point. We'll definitely take it into account.
But we should move on to there are more questions that I'm unfortunately not able to get to, so take a look there. But we should move on to the actual discussion between everyone, bring together everything that we learned from these different talks. Okay, so I warned you that I was going to ask this, and while I'm talking, please, people in the audience, throw in your questions for the panel also. Okay, what do you see as kind of the big goals in microbial diversity? What are you really hoping we figure out?
Well, I can tell you one thing the brewers hope we're going to figure out, which is kind of a practical application. One thing is, how do we make these communities de novo and make them stable? So instead of letting random wild things contaminate your wort and create something amazing, is there some way we could control those communities and repeat them the same time, such that somebody making one of these interesting mixed fermentation beers can do that with the same amount of certainty that it's going to work out as somebody who's using these industrial clonal strains? And that's a really hard problem, I think.
So, I can say, but my long-term goals are all related to one of my obsessions that I don't know how to measure fitness for real things in the real world. And I'm trying to, look at ways to make progress towards disentangling what are sort of processes that are going on in the wild and what are their statistical properties from things that have to be explained by the effects of natural selection, because in the end, you want to be able to sort of say something about that just from the kind of data that you can get from things that live in the wild, not just something that you put in your lab. So for me, this is sort of a goal, but I always see it sort of receding past the horizon whenever I think I get closer to it.
Well, I think in my case, the biggest motivation is like conceptual or theoretical, and is like the foundational question I mentioned earlier. So why there is diversity at all? Why is it so big? If it is big, I mean, how do you measure it, and how it is maintained over time in terms of processes, right? And it's from the theoretical point of view, there are endless series of papers discussing this problem, and for my personal motivation, I become closer to the microbial world because it seems to me that is one of the settings where it's most likely to be able to at least get closer to an answer of to this question.
I mean, I can answer. Daniel Fisher just asked something, and I want to make clear that I do not believe fitness is well defined. That's exactly the problem. I mean, but we still want to be able to measure effects of natural selection, or be able to define what is the effect of natural selection, I think. Or maybe you have to give up on that altogether. But I mean, I would still think that you would like to have some way of saying something about what is natural selection doing to these genomes.
Nandi, I see you're answering some questions in the chat, but actually this gets to another question I wanted to ask, which is, okay, so what measurement would you like people to do, right? Like, what do you think? You know, we're sequencing a ton of stuff, and in my opinion, we're wasting most of it on sequencing people who are basically all the same. What should we be sequencing? What experiments should we be doing? You know, Daniel Fisher said, okay, should we be just sequencing individual microbiomes way more deeply? What do people think?
I think that longer read sequencing, which is definitely happening. Folks at Stanford, for example, are working on really nice technology to look at longer range linkage, plus more time series data, deeper time series sample, and also potentially spatially resolved data. These three ingredients, I think, will improve our resolution greatly to fill in the holes of our models of ecological and evolutionary forces shaping the microbiome. Linkage, obviously, Erik shared some really exciting results. I didn't get a chance to speak much about recombination in my own work, but we also see similar patterns of phylogenetic inconsistency and a lot of recombination occurring. And it's, I think, a really wide open field as to what's happening. And longer range linkage can show deviations from expectations at long length scales that you might not see at short length scales of just 100 base pairs.
I'd like to see more causation experiments. You know, that's one thing that's been frustrating for us is we do all the sequencing to characterize these populations and see what's there. But at the end of the day, you know, knowing that there's an extra copy of chromosome 5 is a little unsatisfying for me. I want to know what genes matter on it. I want to know how they matter. What dimensions of fitness do they impact? How do they create a strain that's better for brewers or worse? So I think getting down to that level of granularity of being able to link genetic cause and effect is, of course, you know, the field's problem, right? But I think it's more acute in some of these population situations where it's been really difficult to do really cleanly controlled experiments testing single variables at one time.
I mean, I don't know how to solve it, but one thing that I think is sort of consistently difficult to get at is the relationship between the sort of the genomic changes that you see and how this relates to phenotypic changes in these organisms. So, and I also don't know how to solve this. But I mean, actually, this project that we started, 8 years ago was sort of trying to get at that, but for reasons that you saw today, we didn't get very far with this. But I still think that that's an area where I'm looking for somebody who comes up with something really clever that you can somehow, in real world situations, look at both these things.
Yeah, I agree. I mean, Erik and Maitreya, I think you're saying pretty much the same thing. And I mean, it's tough because there's so many genes, and they're all changing. They're all changing lots of things in the phenotype. I hope that someone comes up with something, smarter, where we don't need to like try and draw a wiring diagram from every gene to the things that it affects, because I don't think we would understand such an object.
I also think that there is a different level, right, which is the ecological role, which is like in a way phenotype, but it's something more, right? It's also related to what other phenotypes are present, right? So there is also that connection, which is not easy to draw. And yeah, not an answer, but.
Okay, so another question that is getting things a little bit more concrete. This came up already in the talks. Okay, so recombination has played a big role, including mitotic recombination. And right there, you see, like I'm making this distinction between the mitotic recombination occurring within genomes and then the recombination that Erik and Nandita were talking about. There's different recombination rates on all different scales. Is there any hope that we're ever gonna be able to understand microbial recombination? What would that even? Do you even see a way towards understanding there?
I wonder if we need to do some kind of like fine scale observations, similar to the way fly genetics was done back in the day, where people were counting crosses, and some really detailed observations on short time scales could potentially give us some data for how to find out the mechanism for recombination.
Well, I mean, I think obviously understanding all the patterns, no, but I think I don't see any reason why we couldn't find out what are the sort of the key variables that determining these rates. Is it geographic, meeting each other? Is it something about the environment when cells are more stressed, they're more likely to allow homologous recombination? Is it really selection actually fixing these events mostly and weeding out the ones that they don't like? I mean, to learn more about that, I think that should be possible. I mean, I'm quite positive about that. Predicting what's going to happen when you, take samples from a lake every month, yeah, that's another story. I'm not so sure that we're going to be able to do that.
One strategy that we've been taking is trying to decouple the mutation rate and the mutation mechanism from the phenotypic consequences and the genome consequences. So we've been using CRISPR to create a double-strand break to generate a recombinant, and then try to assay that as a type of mutation, and doing that in like a pooled fashion so that we can survey whole chromosome arms and tell like what genes matter and how does it affect the growth of that strain. And so that at least abstracts one step of it out. So you're not trying to solve the entire problem at once, but you're focusing in on one aspect of it. And I think, the same thing is true for a bunch of these species, where now we can start to work and do genetic screens in them, and even in the context of small communities that are constructed, and bring some of those tools of a classical geneticist to some of these more complicated scenarios.
Right, yeah, because usually what we're doing is we're measuring kind of the consequences of a mixture of different processes. The things have to meet in nature, they have to recombine, and then they have to have a pretty fit recombinant. that leaves ancestors, and you're all saying, okay, here, there is a way forward where we can break these down and study them, at least some of them independently, and see what are the key drivers. Okay, and then more generally, okay, so I'm a theorist. I feel like our kind of classic population genetics theory was really built for big organisms that you can look at. And sometimes, like recombination, I think it's not very well suited for microbial populations. So I want to ask, are there any things that you actually think are useful that come over? And are there any other things that really need to be thrown out? And I see here in the chat that Dan Fisher suggested drift as one of the things that maybe should be thrown out in microbes. So what do people think?
Well, obviously, a lot of mathematical population genetics is math. It's saying if this and this and this holds, then that and that and that. And so that's always going to be true. It's just the question is, and so it constrains at the kind of what theories can you make up for explaining what goes on in your data. Now, the question is, are many of the sort of favorite tools from that math very useful? People that have heard me talk before know that I'm very skeptical about this. So I would be with Daniel there. It might be really that looking at the microbial evolution on the timescales that we're looking at is a bit like doing the elephants, but now doing the sort of whole fossil record timescale, right? There things also come and go all the time and so on, and it's not really, drift, right?
I think that we're in an era of measurement. We have so much data, and we don't yet have the statistical tools to see different properties of evolution in the microbiome. And so, for example, take recombination. We still don't have long-range linkage information. So how can we really tell what the true picture of that looks like? We still need to measure it in different ways. And also our statistics need to maybe evolve to catch up with the different types of data that we have. This measurement and comparison with existing models is illuminating because it allows us to see what gaps there are in our knowledge and what fits and doesn't fit. And I think that's actually really useful because we actually have a baseline to start from. And then we can iterate and make new inferences and new models.
Jacopo, I feel like that relates to the things you were talking about at the beginning of your talk with the high dimensional data sets that we need to figure out low dimensional representations of.
Well, yes. The point is always how do we cut our data, right? What do we leave out? But I tend to agree with Erik about the issue of scales when making the comparison between E. coli and elephant, not just the scale, the physical scale, but also like that it's not completely obvious what is the time scale and the spatial scale, and if we're looking at the ecological context we are comparing. So that's essential to keep in.
Yeah, so one of the concerns that I have is like, okay, say I buy Erik's argument that okay, the clonal phylogeny is basically lost in the E. coli did, and I do. Now the question is, okay, we've got this enormous high dimensional data set, right? Big matrix of strains by genomes. And I'm kind of like, okay, how do I start looking at this enormous matrix? I don't feel like kind of classical population genetics gives me too much guidance. There is some stuff where you could say I'm going to run structure on it or admixture or something like that. I don't particularly believe that those results would be so informative. So I think we're at a bit of an impasse where we need some kind of theory to understand how to simplify down our data to the point where we can look at it, but we need new theories, and the only way to generate those theories is going to be from the data. So it's a little bit of a chicken and egg problem.
I think one thing, I mean, this, and we still have to finish this paper, but is that, so I talked only about the core genome of these guys, but I mean, it's very clear if you look at gene content and turnover of gene content, that I would think that that's the first dimension that is the most important. Okay, so the main differences between these strains are in the set of genes that they have. And then second, the alleles that they carry, which they mainly switch through recombination events, right? And so, you know, it's not a lot of progress, but I think it's, you know, it's a start, right? I think to first order, the changes are controlled by changes in gene content, which come from recombination events. But I think those are the main, most likely to have phenotypic effects.
And I just wanted to say about this comment about the data being really complex. We have this huge matrix of genotypes versus species. Well, we can start to analyze these species one by one. Like take, for example, Erik's study where he looked at E. coli. He didn't need to consider all the other species in that lake to make some really interesting insights. And he also applied some standard statistics like S per base pair to make some really interesting insights about how recombination deviates from our expectations. So we can actually simplify the data, and we can actually use what is in our toolbox to come up with new theory. And I think that potentially is a way forward while we start to figure out how to put all the pieces together across species.
I think so. Okay, so let me just close by asking, are there any questions about microbial diversity that you think we're, you know, poised to really make big progress on? And maybe I'll say, try not to just answer, you know, your own area. Obviously, you do think that, that's why you're working on it, but maybe something else too.
Well, I think it's really been interesting that one thing across multiple talks today has been the gene flow between and within species, and how that can make big jumps in how these populations change, and whether that's from a mating of two species to make an interspecific hybrid, or whether it's from a horizontal gene transfer of small numbers of genes. The consequences of that are something that I think is starting to become more clear as we start to appreciate who's exactly in all these little genomes, as opposed to looking at them in mass.
I mean, I mean, maybe it's not very new, but I have the feeling that there is a shift going on, at least from the way I see people thinking about things, from sort of people thinking of evolution happening in some sort of environment that is given almost as a sort of a background that is set, right? It may change within time and so on, but it's a set, it's separate from more in the direction that it's all in the interactions between the individuals in the community, right? And that's maybe the dominant term. And so I have a feeling things are sort of, that's becoming more and more sort of accepted, or at least more and more people believe that it might be what's going on. But you know, you never know breakthroughs before they happen, no?
Yeah.
Jacopo, Nandita, any areas?
Well, I tend to agree with Erik. I think that, I mean, as I said, I think we need, I sort of highlighted some three ways to deal with these high-dimensional data in terms of approaches. And I think that where this answer will come from is to combine these different approaches, to combine also like smart experimental systems where one can really look at, can really control the conditions and can perhaps have much higher diversity than what is currently possible with the current experimental settings. So I think that that is, because that is in a way the only way to really draw a causal link between mechanisms and observables, right? Otherwise, we will always deal with, well, I observed this, I also observed that, I also observed something else. And this is one explanation of, with some errors in it. Right? So manipulating is essential in that respect, I believe.
Yeah, I agree. We need to have a lot of crosstalk between what's going on in the lab, in a test tube, versus experimental evolution in a mouse, for example, and of course, observing natural populations. And we learn a lot from each level of granularity. We also have trade-offs. In the natural populations, we can't really control everything, but we can in the test tube. But then we lose the complexity that we actually observe in natural populations when we're looking at a test tube. Maybe the happy medium is mice. So it's a really exciting field, and I think that what we learn from one setting will definitely inform the other.
Well, we're well over time at this point, so thanks to all you for the great talks and great discussion. Thanks everybody for tuning in, and to Tierra Ward for handling all the technical side. Remember, next month workshop is Taking Biological Theory Seriously. Please nominate your favorite papers that quantitatively predict real data. And easiest way to keep in touch is just to go ahead and subscribe to the YouTube channel, so that the Emory TMLS YouTube channel, so you get all the updates. Thanks, everybody.
Thank you.